So thank you for the introduction. I'm Birthe from KU Leuven, and this project is in collaboration with my promoter, Tom Schrijvers, and Peter Dedecker, who is a professor in the KU Leuven Lab of Nanobiology. So today I'll present to you a nice and perhaps unexpected application of domain-specific languages, namely in the domain of fluorescence microscopy. So let me first walk you through the application, the principles of fluorescence, and what fluorescence microscopy actually is. So microscopy techniques can be used for different studies, usually to study interaction or the behavior of cells and tissue. And the type of microscopy that we use is dependent on what we want to see or monitor. But today is about fluorescence microscopy, which is mainly used to study DNA. And I think I have to give you a bit of biology here. DNA. So DNA can be found in two parts of the cell, mainly in the nucleus, the green big part in the middle of the cell, but also within the mitochondria, which are these smaller structures here, whose main task is to transfer energy in the cell. And DNA studies are important. They are used for cancer research. So one of the most used cell lines is HeLa cells, which are cancer cells, but also in recent years for the development and discovery of COVID-19 vaccine, fluorescence microscopy was one of the mainly used techniques. So with fluorescence microscopy, we can look at different scales, mainly at the micrometer scale, for instance, to look at bacteria, mitochondria, or cells in general. But with some specialized techniques, we can also look closer at the nanometer scale, for instance, to study proteins, viruses, and also DNA. And these fluorescence images are pretty. I find them pretty. And what you see here, remember we wanted to study DNA in the nucleus and the mitochondria. So what you see here is the nucleus being colored in blue, mitochondria in green, the left picture. And what we see in the back is what we call the cytoskeleton in purple. Also on the right, nucleus is blue, mitochondria are orange, cytoskeleton is green. Now, how is it possible that these different structures also have different colors? That's because we can label them with so-called fluorophores. And fluorophores are kind of fluorescent proteins. And in practice, this means that we go to the lab, we have a pipette, and we add them to the cells. Then we have to wash the cells and respect a certain incubation time, and then they become fluorescent. So if we put them under a microscope, they will start giving a very bright color. And these fluorophores, or fluorescent proteins, are not as exotic as you might think. We can find them in nature. For instance, here, the GFP, or green fluorescent protein, is the most well-known one and can be found in jellyfish. Also, in the sea, we can find DS red, which can give a red color in coral reefs. But also in your drinks, we can find quinine in your tonic water. Maybe you know the experiment. If you have black light, which is actually ultraviolet light, and you shine it on your tonic water, it starts shining blue. That's actually what fluorescence is. So this is the spectrum of visible light. And it ranges from purple to red, and each color corresponds to a certain amount of energy, also reflected by a wavelength. So, for instance, the purple light has a shorter wavelength, so a higher amount of energy than, for instance, red light with a longer wavelength, so less energy. So what's important here is that ultraviolet or violet light has higher amount of energy than blue light. So that will be important for tonic water. Because if we shine this ultraviolet light on our tonic water, it doesn't shine violet, but it's blue. And in order to explain actually what the principle behind that is, fluorescence, I will show you a so-called Jablonski diagram. So the tonic water with the quinine is in kind of ground state, ground energy level, and it gets excited with ultraviolet light, which is the purple colored line here. So it is excited, it goes to a higher energy level. When it then falls back to the ground state, it will start shining a different color, which has less energy. So that corresponds to the longer wavelength So that is blue light. So we have purple for excitation and blue for the emitted light. And that's what we call fluorescence. And if we put fluorescent cells under a microscope, the concept to do so is actually quite simple. So we start with some emission light of a certain color at a light source. This light source is usually a laser or a LED. And this light passes through a filter. This filter has its purpose to filter out only monochromatic light. This means that we will have only light of a single color or a single wavelength. We filter out noise, other colors, to have a single excitation color light. It then goes to a dichroic mirror, and dichroic mirrors are a special kind of mirrors that reflect some wavelengths of light and let other wavelengths pass through. So if you follow the arrows, it is reflected by the dichroic mirror, goes through the objective to the cells. And if these cells were fluorescently labeled, they will start shining a different color. It's what I visualized by the red arrows. So the fluorescence light goes back through the objective, now through the dichroic mirror, through another filter. This filter again filters out different colors, noise, reflections, and so on. And what we actually want to detect is this fluorescent light, which we do by a detector, which is usually a camera or you looking through the eyepiece of your microscope. So this is the concept of fluorescence microscopy. But in practice, a microscope looks more like this. So it has some more sophisticated components. But the principles are the same. We start at a lamp here. So this is where the excitation light comes from. It goes to the objective, to the cells, which are here, and the cells start shining a different color, now red, which goes to the camera or to the eyepiece of the microscope. So conceptually, it's the same thing, but we have some smart components in the microscope. For instance, these are filter wheels. So we don't have a single filter, but we have a filter wheel, and if we turn the wheel, we can filter out a different color. Also, we have this thing, a motorized stage. It's not always motorized, but it's a stage, a kind of platform on which we can place the cells and which can move in X, Y, and Z directions in order to look at different positions of our sample or to get a different focus in our cells. Now, fluorescence microscopy is very much used both in industry and in academia. And you can see this by popular microscope vendor brands such as Thermo Fisher, Nikon, Olympus, but also by companies, mostly in the pharmaceutical sector, which use fluorescence actually in their everyday tasks. For instance, when in drug development for their control tests. So I've named some examples of Galapagos, Johnson & Johnson, Roche, which really use this technique every day. Also in industry, it's used in the agricultural sector, for drug development, for food in the food industry, so it's really everywhere. Also in academia, there are many publications about fluorescence microscopy, but also using fluorescence microscopy, evidenced by many publications in journals like Cell and Nature, which are, I think, many known journals with an impact factor of about 70 in 2022. So fluorescence microscopy is important. Now, let me guide you to one day in the life of a fluorescence researcher. So a fluorescence researcher is usually trained as a biologist or a biochemist, but he or she also has to operate the microscopes. So we start the day with cell preparation. We have our cells, we have to label them. So we go to the lab, take our pipette, start labeling them, we wash them, and we wait for it to incubate. And actually this process may have to be repeated a few times, or it can also take multiple days. But when the cell preparation is ready, we start our microscope setup. Yeah. So what we have to do before setting up an experiment is to choose the appropriate filter in our filter wheel, to choose a dichroic mirror that is compatible with the wavelengths of our fluorescent protein, which we labeled the cells. We have to move the cell to the sample to get a good position to look at. We have to find the focusing distance and so on. So imagine that a researcher has to do this manually. I think it's still doable for setting up this. We have to just manually turn the wheel, put the cells in a different position. That's okay. But during the experiment, these settings might have to change. So we have to move the sample again, turn the wheel again. And also these experiments can sometimes take up to 48 hours. So we might have to do that multiple times, stay focused. Also detection of some phenomenon in fluorescence microscopy is happening very fast. So it can sometimes be less than a second for something to happen, which you want to capture. So I think the conclusion is that it's almost impossible to do this manually. For a very long time, doing things in different time intervals and do it very fast, it's impossible to do it manually. So the conclusion is we need software. And the software should have a few requirements. It should be able to change the filter wheel, to activate the light source at a specific wavelength, because that might also have to change the excitation light color, do the detection with a camera, making pictures, to move the sample over different positions, ideally also in the different directions, X, Y, and Z. It should be able to repeat the program for a number of times or at different time intervals. And ideally, it also has a graphical user interface. And software packages are available in industry, but the needs in industry or academia are actually very different. For example, in industry, software is mainly used for repetitive tasks. Think about these control tests in the pharmaceutical sector. It's all day long, it's always the same process, so it's repetitive. Whereas in academia, we want to have the freedom for creativity in the experiments that we set up. So we make custom experiments. Also in industry, what we often see is that these software packages come together with the microscope. So it is one package, microscope and software together, which are usually provided by the vendor of the microscope. So it's often tailored to specific hardware and vendor specific. But in academia, what we often see is that people build their own microscopes. They also change the parts that are in this microscope. And they also want to have the freedom in configuration, which might also evolve as the research needs evolve together. So, conclusion, the needs are very different. So what we actually like to have is something that fulfills the software requirements, my previous slides, that allows for custom experiments so that we can build experiments that are arbitrarily long and complex, just like we need them to for our research. And we want to have something that is modular in terms of hardware that it supports. So today I will present you a domain-specific language to fulfill these needs. We named it Fluo, and it actually consists of 3 parts. It has a measurement controller, which is actually the core, the domain-specific language. But it also interacts with a graphical user interface, and it connects to different kinds and brands of hardware. So the domain-specific language is written in Haskell. And one of the advantages is that we can choose the syntax. So the syntax is actually very similar to the software requirements that we saw earlier. A program is defined as a list of measurement elements, and these measurement elements are like the software requirements. So we have detection to activate the camera to take a picture. We have irradiation, which will activate the light source at a certain color for a certain duration. We can wait for a couple of seconds. We can repeat the program. We can also do time lapses and stage loops. And actually, the time lapses and stage loops are nice to have. They are not always available in other software packages, because what's nice about them is that they nest another program, so we can now freely nest programs in order to construct custom experiments that can be arbitrarily long or complex. Also, the semantics is kind of straightforward. As we have a list, we can just use a fold map. And executing a measurement element is also activating the camera, activating the light source, introducing a thread delay, and so on. So what we see actually is that on the programming language side, it's not very exotic. It's simple what we use, but on the domain side, it makes it different because they can nest programs, they can make custom experiments. So what we also wanted is to be modular in terms of hardware. And I took an example of this motorized stage. So what we do is we use type classes, for instance here for a stage, that we can implement to support a different brand. So for instance, Marthauser is a brand of stage, and we just implement this type class, and it is supported in our DSL. So we also have a prior stage, which is just a different brand, and even a remote stage, which can adjust its position by itself, by seeing what it monitors. Also, it's modular in terms of the microscopes that it's deployed on. We have 3 microscopes in the lab of a different brand on which it is deployed. So it is modular in terms of hardware. What sometimes happens is we get new hardware components, we order them, and these usually come with a DLL written in C code with which you have to connect. So what we do is we write a different type class for it, and we have to be compatible with the DLL that is provided, which is not always as easy, but doable. So third, we also have a graphical user interface, and this GUI is written in Igor Pro. Igor Pro is software that is often used for data analysis in a biological or biochemical context. And what we do here is we have constructed some buttons, and by touching these buttons, the users can construct their programs. They also see them being built, and by using these buttons, we make sure that the users, who are not programmers, they are biologists or biochemists, always construct syntactically well-formed programs. The only thing that they do have to know, so they need to have a notion of indentation, so that also the program will do as they expect to make well-behaving programs. Okay, so let's look at an example program. This one was constructed with the buttons in Igor Pro. So we repeat 5 times an irradiation of 2 seconds using a light source that is called lumen core with violet light at 15% power. We wait for 3 seconds and acquire an image with the camera. So we repeat this 5 times. We wait another 10 seconds and require a final image. And if we now see how this gets translated to the domain-specific language, again, I think it's not very surprising. It's a list with some nested program, which is again a list. So again, on the computer science side, not very exotic, but on the domain side, very much of a difference. So let me get you to a specific use case in the Nanobiology Lab of KU Leuven that would not have been possible without the use of the DSL. For this, we needed specialized or more advanced time-lapse experiments. And what we can do with this is simultaneous FRET measurements. So let me try to explain you what that is. FRET is the abbreviation of Förster Resonant Energy Transfer, and is a kind of energy transfer between 2 molecules that are in close proximity to each other. And close means less than 10 nanometers. We use FRET to study the interaction or the relative orientation between these 2 molecules. And in practice, this is often used for example, DNA mutations. So what usually happens if we get a molecule and we have excitation light, we shine on it, and it starts emitting a different color at a different wavelength of light. Now, if we add another molecule that is close enough to the original one, it will start behaving as a donor, D, and an acceptor, A. So we excite the donor, it transfers its energy to the acceptor, and the acceptor will start emitting light. And choosing a compatible so-called FRET pair, so these 2 molecules together, will give us insight in the interaction between those to study DNA mutations. Now, What we want to study this is the so-called FRET efficiency, which is the fraction of the emitted light of the acceptor and the excited light of the donor. But what is actually very challenging is if we have different FRET pairs working on different structures, and we want to measure these different FRET efficiencies at the same time. So we want to measure multiple FRET pairs simultaneously. And this is what my colleagues in the lab have developed. It is a specialized protein that allows us to do so. And in the experimental part, what we need is to take 3 different pictures at different time intervals. So in an on and an off state, and we need to take 3 pictures, one with a color, an excitation color that is optimal for donor excitation and donor emission, one that is optimal for donor excitation, acceptor emission, and one for acceptor excitation, acceptor emission. The middle one is called the FRET channel because we excite the donor, it passes its energy to the acceptor, and the acceptor starts emitting light. So here I have a code snippet using the DSL that was actually used during one of these experiments. So what we see is a time lapse, also a stage loop, because we want to look at different positions. And then these 3 pictures. So we irradiate at different colors, and we take a picture each time. So this corresponds to the DD, DA, and AA picture of each time point. All right, so to reflect, this DSL has been used daily in the Nanobiology Lab of KU Leuven since 2015. And it actually not only increased the operator efficiency, but it also allowed the an increased expressivity of experiments. And this is evidenced by novel experiments like the FRET measurements in the lab. As it was developed in Haskell, the users find it a robust DSL, so they trust the software. So that's another improvement. What was difficult is the steep learning curve of Haskell. So what was not always trivial is to understand the laziness, for instance, when we're taking pictures. And also the connection with C libraries, for instance, when we get new hardware components, is not always easy. But I think to conclude, on the programming language side, I think it is a simple DSL. It's a nice application of how we can use simple structures that do really make a big difference on the side of the domain experts, which are in this case biologists or biochemists. Okay, thank you, and I'm happy to answer questions.
Hi, very interesting, first of all. So I'm not a Haskell expert. In fact, I know almost nothing about Haskell. So maybe some of the code you showed was Haskell and I just couldn't tell. At the very end, you mentioned that there is a steep learning curve due to the use of Haskell. So the code that you showed us seemed to be, quasi-English plus a little bit of programming. So was that Haskell? And if so, can you talk a little more about what the challenges are for domain experts to use it?
Yeah, so maybe I should go. This is not Haskell. This is the program being built with the DSL. This is Haskell. What was difficult, so maybe first I have to explain, we have 3 parties involved here. We have the real domain experts which are using the DSL. And then you have the collaboration between me and my promoter, which are on the computer science part, and the professor of nanobiology, who is in nanobiology. So it was him who first started building this, and in collaboration with us, we started to exploit DSL features and so on. But for him, he was the one who had to learn Haskell. And it is, for instance, not always trivial to understand the laziness. When you take a picture, you don't immediately use it, but you want to take it now. And I think on the computer science side, it's trivial. We will use monads and we will do stuff with that. But for him, it was difficult to understand. That's one example. Also, he started with the development of it, but not exploiting the DSL features yet. So it was in collaboration with us that we started doing so. And also, with this application case, I found some interesting topics on the really technical side in my PhD, on which I worked, which can then be used again in this application.
Thank you. Do you think this approach could be modified and applied to scanning probe microscopy, like atomic force microscopy or things like that, or also something else where you move a stage and have to control things, or things like 3D printing? I was wondering if there's any way you could take this work and apply it to those other domains.
Yeah, thank you for your question. I think it's a good question. It's always a difficult trade-off between having a domain-specific language, which remains domain-specific, and something that can be applied to different domains. So I believe that it can be used for different microscopy techniques because the principles are similar. We will maybe have to add stuff. I don't know if it's nice to also do it in, for instance, 3D printing, the example, because then it's a different application domain. And maybe it's nice that we don't also change the syntax, sometimes the behavior. So I think that we want to have a domain-specific language and to have it remain domain-specific, which is in this case fluorescence microscopy.
Thank you.
There's still a couple of minutes. Other question?
So how does the pipeline look like? So you showed the code that you write in the DSL and the Haskell representation, but what's the connection there? So do you write the DSL code in some file and then that gets parsed into the descriptive and then you execute it, or what's it, the pipeline?
Yeah, so in the graphical user interface, we constructed and they're connected, so almost one-to-one connected to the syntax in the DSL. And then via the DSL, we connect to the hardware.
Mm-hmm. Okay, so you never actually write the code yourself. You basically just use the user interface to construct it.
Yeah, so that depends again on, so these three parties, the real domain experts, they don't like the idea of programming. So they use these buttons, they use the interface. But for instance, if I want to construct a program, or some of my lab members that do like programming, they will start to use a terminal, which is also possible. But it's mainly about the idea of programming. If you're not trained for it, you like the interface more.
So let's give the speaker a round of applause.